So what Trim-O-Matic does can be divided into two different approaches. So it trims the data and it filters the data. What is the difference between filtering and trimming? Well, when you do filtering, you're going to remove whole reads from the FASTQ file, so complete reads. When you trim, you are going to remove bases from the FASTQ file. So filtering, you remove reads. Trimming, you remove bases. So, for instance, adapter dimers, they are filtered. You remove the whole read. If the whole read is adapter, you're going to remove the whole read. But you can also trim low-quality bases from the end of your reads. That's trimming. So what are the steps that you typically do here? Again, it depends on what FastQC is telling you, but you remove adapter dimer contamination, obviously. When do you do this? When you have a lot of adapter dimer contamination. So when 10% or 14%, as was the case in our file, or 20% or even more of the FASTQ file consists of adapter contamination, you are going to remove it. The main reason for doing it is to speed up the mapping. So the next step that we're going to do when the quality has improved is the mapping. Aligning the reads to the genome sequence is very slow. You can imagine that. You have to align 8 million sequences to the full genome. That can take a very long time. So you're going to make the mapping as efficient as possible. So, and when 20% or 15% of your FASTQ file consists of adapter dimers, you're going to remove them because they will not map. They will not map to the Arabidopsis genome. They are not from Arabidopsis. But the mapper will try. He will try to map them. So he will lose a lot of time trying to map these adapter dimers that will not map. Now, when you remove adapter contamination, the tools that do this, they will automatically also tackle this kind of contamination. So in your FASTQ file, you will have adapter dimers, so reads that consist completely of adapter, but you will also have this case. So when you select fragments for your library, you're going to do a size selection there. But these size selections are never perfect. You typically also select fragments that are smaller. So it can happen that the size of the fragment is smaller than the size of the reads that you're generating. So when that happens, you get this. You get a read that is part fragment, but also part adapter. Of course, you can imagine that they also will be very hard to map. They are part fragments, so part of them will map to the Arabidopsis genome, but the adapter part will not map. So the best thing to do here also is to remove the adapter contamination. And the tools that do this, that remove this, will also remove this. Because it's basically the same thing. What will they do? They will take the adapter sequence, they will align it to your reads. If your read matches the adapter sequence, they are going to cut it. They can also trim low-quality bases from the ends of your reads. You will have a tool that can remove reads with a low average quality. The tools can be used to remove poly-A tails from RNA-seq data. Normally your sequence provider does it, but and you can use these tools to do demultiplexing. So what is demultiplexing? Well, it happens when you put multiple samples in the same lane. So to save costs, they often do that. They put multiple samples on the same lane. But then, of course, you need to include a barcode in your adapter that will tell you for each read, for each fragment, which sample it comes from. So that after the sequencing, you can split up your reads into one file per sample. So this splitting is normally also done by the sequence provider. So what the the base calling software generates is one huge FASTQ file per lane on your flow cell. If there were multiple samples in the same lane, he has to demultiplex that FASTQ file and split it into one FASTQ file per sample using these barcodes. Now, I have seen data where the sequence provider has not done it, so you have to do it yourself.